Hello students, welcome back to another class, and today's topic is DNA sequencing. Yes, we have already completed about the genomic libraries, cDNA libraries. We have seen in depth about those topics. Now is the time that we switch on to DNA sequencing and its methods. Okay, so now the main purpose of genomic library is not only to conserve the DNA, but at the same time the main purpose is also to sequence the DNA, especially genomic libraries. But many different strategies has to be made to sequence that lot amount of genome at once. So before sequencing that huge amount of DNA, let's see that in previous times how the DNA was sequenced. Okay, so in around 1971, first phage DNA, that is bacteriophage, this Psi 174 was sequenced. It is around 5,000 something base pairs, 5,800 or 5,300 something base pairs. Then in 19 again, in later stages, the human mitochondrial DNA was sequenced. Okay, so this is how the sequencing started before, and Sanger's was the main scientist in doing all that. So let's first study about the Sanger's method. Sanger's method is also known as chain termination method. It's very important. It's also known as chain termination method. Now these methods are very logical methods. Now let's see what happened. Suppose that you have a DNA of interest. This is 5 prime to 3 prime, and you don't know exactly what is the sequence of this DNA. Now, first of all, this is a replication based method. Replication based method. Okay, the Sanger's method is also known as chain termination method, and it is a replication based method. Now because the replication starts from 3 prime to 5 prime, so let's draw like this. It's 3 prime to 5 prime, and this is the fragment you want to know the sequence. First, what you will do, for replication, primers are required, so you will add primers. What you will add? You will add primers here. Now point is, you don't exactly know what is the sequence of the DNA, so how can you add primers? So what you do, because this is a double stranded DNA, initially there is a double stranded DNA. Okay, so first what you do, you don't add primers, but I'll write plus here. So first you do denaturation. You denature the DNA into single stranded form. Now the problem is that you don't know about anything about this DNA. Then you clone this DNA into a M13 vector. Yes, you clone this DNA into M13 vectors. And in M13 vectors, you only add one type of primer, that is forward primer, that is 5 prime to 3 prime. Now, if the DNA of interest is large, you want that the sequencing should be very fast, so you can add primers from both the sides, but still you have a single stranded DNA right now, so it's no use. So 5 prime to 3 prime primer you have added, and for what? You have added the primer for specifically for your M13 vector because the vector sequences is known. So this primer will go and bind here, and you add another important enzyme known as DNA Pol 1, but not full, Lenau fragment only. It's very important to note that here we cannot use full enzyme because it has 5' to 3' exonuclease activity, and we don't want that. We want Lenau fragment, and you will add DNTPs that are radio labeled by P32. Right now, nowadays P33 or sulfur labeled is also used for ease. Okay, so P32 DNTPs radio labeled you have added. And finally, the main recipe of Sanger's is DTNTPs. Okay, so from here you will divide the reaction mixture into 4 different aliquots. You will divide into 4 different aliquots. So this is test tube A, this is test tube B, this is C, and this is D. Now why we have created 4 different test tubes, you will understand in a moment. You see that in the test tube A you will add DD-ATP. In the test tube B you are adding DD-GTP. In third you are adding DD-CTP. And in the fourth you are adding DD-GTP. Now see, other things are same, plus you will add this Lenau fragment DNTPs that are P32 labels and your this gene of interest or vectors. Okay, other things you will add as it is, but only thing that you will differ is your dideoxy. Now what is dideoxy? If you see the structure of nucleotide, so DNA and RNA has a one important difference. This is your 2', this is your 3'. So both the DNA and RNA has a little difference. The RNA has OH on both the sides, and DNA has 2' deoxy. That means instead of OH it only has H. DNA. Okay, and RNA has OH on both the sides.